Okay, well, thank you everyone for coming, even though it's San Jordi. Miguel Rubí agreed to give a talk today, and I think this is a very good thing. I'm very thankful. Miguel Rubí, I don't think he needs a long introduction. He's been a professor in the department for as many years as the department has existed, and for a long time in the previous department of fundamental physics. And now he's going to talk about self-assembly in biological soft matter structures. So please, Miguel.
Yeah. Thank you, Antonio, for the introduction. I would like to give you some ideas about a very important process taking place in soft matter and in biophysics, which is the self-assembly. You all know what self-assembly is. It's basically the formation of structures of different types from a disordered set of building blocks, disordered set of particles or entities. But what makes the difference is to treat this process in equilibrium, because it was first, we started to analyze it in equilibrium. So when the system, the particles or the building blocks interact to form the structures, and in a non-equilibrium. In non-equilibrium, you add some substances, you apply some external gradients, and you apply some external forces, and so on and so forth. And then the kinetics of the self-assembly process in equilibrium and outside equilibrium is very different, and the type of structures is very different. And it requires, in outside equilibrium, it requires a more deep study because the system is coupled to the medium. So when you have to modify the structures through the medium, and then in that case, yeah, you have to incorporate the medium in the analysis. The work I'm going to present to you was started some few years ago, made in collaboration with one of my students, Andres Arango, who has a fellowships in my group, and Professor Daniel Barragán from the University Nacional of Colombia. Okay, yeah, this is the outline of the distribution of my talk. First of all, I'm going to give you some general aspects about out of equilibrium self-assembly. What are the differences with respect to equilibrium self-assembly? I'm going to discuss two particular situations, two particular examples. One is the formation of gels, and then the second one is the formation of patterns, of Lisigan patterns. Lisigan patterns are formed because of the interaction of two electrolytes, and gelations is formed because of the when you add a chemical substance to another one, and then there are some fibers that come up, and the fibers interact and form a network, and this network is a gel. Well, I told you before that there is a crucial difference between equilibrium and non-equilibrium self-assembly. In the case of equilibrium self-assembly, there is a criterion for the appearance of the structures. So the structures appears at the minima of the local minima or global minima of the free energy, or the thermodynamic free energy, we all know. However, when you are outside equilibrium, there are several experiments indicating that structures are not formed at the minimum of the free energy, right? And then the free energy, the thermodynamic free energy, which is basically a chemical potential, a thermodynamic potential, which excludes dissipation, right? Is utilized to characterize ideal processes in the absence of dissipation. When you are outside equilibrium, dissipation is so important and it's crucial that this free energy is not able to characterize the structures. And I'm going to present a new criterion, proposing a new free energy functional, an effective free energy functional, which takes into account all the characteristics of the processes, and it can be used to detect the formation of self-assembly structures outside equilibrium, quite generally. So as I said, self-assembly is the organization of discrete elements into structures. And an assembly of structures is an arrangement of building blocks. You can see this in this picture. So the blue particles, colloidal particles, are inactivated particles, and the yellow particles are activated particles. And when the particles become activated, they can form these structures. So to pass from the blue particles to the yellow particles, you have to activate these blue particles. And the activation is made by chemical substance, right? So in the absence of when you don't add any substance, when you are in equilibrium, the particles cannot be activated, and the structures cannot be formed, right? Yeah, you can see well, details and references and yeah, more information in this article we published in the physical chemistry, chemical physics, in the section perspectives. There's a kind of review, and then you can see more details about this, the process of the equilibrium self-assembly. Well, this is a typical out of equilibrium self-assembly. We start from a set of building blocks in blue, particles. And then you had to activate the particles because, as I said, the blue particles are unable to form structures. You had to activate them, and then for this you add the fuel, and then the fuel makes it possible that the particles become activated and pass from blue to yellow, and these yellow particles can form the structures. And when the fuel is consumed, right, these structures fade away, and then they come back to the initial position. Right? This is basically an example of an illustrative example of the process of out of equilibrium self-assembly. And it's out of equilibrium because we have to add fuel, right? We have to add energy, we have to add a mass in order to promote the process, the self-assembly. So this non-equilibrium self-assembly a process leads to the formation of stable, metastable, stationary, kinetically trapped structures, and is mediated by dissipation, right? So this energy taken from the fuel is dissipated in the formation of the structures. It disappears, and more energy has to be given to the system in order to continue the process, okay? And non-equilibrium self-assembly process is an important point that they are the previous steps in the formation of self-organized structures, of more ordered, large-scale structures. So in the same way that nucleation is the first step in the first transition, and non-equilibrium self-assembly is the first step in the formation of self-organized structures. Well, these are examples of structures. There are many microtubules assembled from tubulin dimers, gels assembled from fibers or microtubules, plant cells assembled from microtubules assemblies, and leaves assembled from plant cells. There are many others. You can also see the cases of self-propelled particles that tend to coalesce and to form these structures. These microtubule-based structures or active gels, you can see three examples here. A is a network, a contractile or a silver structure. The B is a microtubule aster, and the C is a vortex. And you can also, I can give you another example I'm going to mention in more detail at the end in the second part of my talk, is the formation of magnetic hydrodynamic assemblies, right? So you have colloidal particles, magnetic colloidal particles under the action of a rotating magnetic field. So the particles, you see that rotate and perturbs the fluid. So the fluid starts to move, the Reynolds number increases. It depends on the strength of the magnetic field. And then there are some structures formed just in the middle. For example, 3 particles are located in the central part, and depending on the condition, you can have more particles concentrated in the central part. This is will be another example. Well, there are important questions on non-equilibrium subassembly. The first one is how the formation of the structure is governed by the law of thermodynamics. This is a very basic question connected with the fact that, as I said, the free energy of the system is not able to provide the description of the type of structures appear in the experiments, and then you had to use different thermodynamic scenario in order to predict the formation of the structures. Energy dissipation is very important, as I said, and the amount of energy dissipated should be calculated because it plays a very important role in the formation of the structures, right? And then we had to provide a theory able to calculate the dissipation and the energy dissipation of the entropy production rate as a matter of fact. And this first point is the point about the lack of a non-equilibrium potential to properly describe the formation of the structures. Well, these questions concerns the theory. There are several computational approaches proposed to analyze the formation of the structures. But these computational approaches have technical challenges because the number of particles coming into play are very, very large, right? And then this complicates the analysis, but even though it can be done, but these computational approaches describes the kinetics but don't describe the dissipation. They are unable to quantify the dissipation. So you have to combine computational approaches with a proper thermodynamic theory in order to account for the formation of the structures. Okay, you can see here, different models used to analyze the kinetics and the thermodynamics. The models can be divided into dynamical models and thermodynamical models. Dynamicals are computational and kinetic. You can identify here the kinetic Monte Carlo, molecular dynamics, Langevin dynamics. And among the kinetic models, the reaction diffusion, magnetohydrodynamics, mesoscopic thermodynamics, and some others. And the thermodynamic methods involve the Gibbs entropy and the information entropy, the minimum entropy production, which can be used when the system is not very far from equilibrium, dual entropy production, and so on and so forth, configurational entropy, and so on and so forth. But these are some models that have been used to analyze the kinetics and the thermodynamics of the process. Well, let me first focus on the first example I want to give you. The example is gelation. So it's the formation of networks of fibers, network of fibers in this form. And then you can see here the different steps of the process. You start from a disordered set of building blocks. Why building blocks? You activate the building blocks adding fuel. Then these building blocks become activated, and they are called A1. So I have Y building blocks of type A1, and since they are activated, they can assemble, right? And then assembles a building block, a fiber containing N-1 building blocks, assembles to another one with to a building blocks, giving a larger fiber containing N building blocks, which can be disassembled and producing another building block. This is a first order. These are called the first order structures, but promoted by a first order activation. But there is another activation because these fibers, these cylinders or rods, can, thanks to a second order activation, coalesce, right? And then they can form the network, right? So this a fiber containing N building blocks can be added to another one containing N-N to give a another one containing M building blocks. So in this way, this network can be formed, right? These are the different steps intervening in the process. Okay. The mechanism is, well, you can see here the names, the chemical substances. The building blocks are the benzoyl cysteine. The activators is methyl iodide. The activated blocks are DBC methyl. There is an intermediate activated building blocks, and there are, as I said, first order structures, which are the linear fibers, and second order structures, which is precisely the gel formed by the agglomeration of linear fibers, right? So when this fuel is consumed, right? When the fuel is consumed, well, these networks disassembles and goes to the initial state. Okay, so this proves that the system is outside equilibrium and there is a dissipation involved. Okay, you can see here some theoretical results obtained from the model I'm going to describe in a moment, and the experiments. So hollow circles correspond to experiments, and the lines, the solid lines, are the results of the model. Okay. Well, where is my arrow? Yeah, and then you see that. Can't see the arrow, so maybe that. Yeah, you see that here. This picture represents the concentration of the building blocks. That you see that I mean, it decreases. I see that the connection is not very good. Well, you see that the concentration of building blocks decreases because I mean, this is the concentration of inactivated building blocks. It decreases because there are more activated, right? And then in the second picture, what we represent is the concentration of the activated building blocks, which increases, right? Which increases, you see? Whereas one decreases, the one increases. And then at 400 hours, more activated is added. So all the building blocks and disappears, and then you have to add more activator, more methyl iodide in order to produce another time the structures. And the third picture shows is a dynamic like scattering experiments, which clearly shows the formation of two structures at this point at 150 hours and 700 hours. And then you see that this is the signature that the gels appears, right? The peak in the dynamic like scattering experiment is the signature that there is a coherent structure in the system. Okay. The model, well, to characterize all these different steps, we had to introduce reaction coordinates. This is a summary of the reaction coordinates used just to characterize all the activated processes. You have an activation, the activation of the building blocks. You have also the activation of, I mean, the formation of the higher order structures and the formation of second order structures from the first order structures. You have different types of reaction coordinates, right? And the idea is to calculate this, to analyze these activated processes. So a self-assembly is an activated, it's a self-assembly process is an activated process. And then the idea is to analyze this activated process from a Fokker-Planck equation for the probability distribution function, which you can see over here, right? This Fokker-Planck equation can be obtained from the energy dissipation, T sigma. Sigma is the entropy production rate along the reaction coordinate. So this picture shows how a substance transforms into another by means of a rate process. And then the Fokker-Planck equation in the space of the reaction coordinate describes the evolution of the probability distribution function, and also the current, the activation rate. The activation rate is given by the law of mass action, right? is given by law. I mean, I describe this activated process, each step of the self-assembly process as an activated process in which a state 1 transforms into another state 2 through a reaction coordinate, and then I use this low mass action, which combines two Arrhenius factors. This is the theory, right? Well, the model, you see here that I had to consider different reaction coordinates, and an important point is that there is a reaction coordinate, another reaction coordinate, which is or another coordinate, which is the orientation of the fibers, the orientation of the fibers, theta n. Theta n is the orientation of the fibers, which depends on time. This is also a variable that should be considered in the set of variables for the probability distribution function. Okay. Okay, once we have this, and then we collect all the reaction coordinates, so the 4 reaction coordinates we have in the process in gelation, and calculate the Fokker-Planck equation, which is given by this equation, and take into account the chemical potential. Here, the psi is an activity coefficient, because the process is not ideal. It's different from 1. 5 is different from 1. P are the probabilities. And then what we obtain are 2 Fokker-Planck equations, one for the first order structures and another one for the second order structures. This is for the formation of the red bars, red fibers, and this one is the other one is the one for the formation of the green networks, right? Both equations are coupled through the boundary conditions. Then you can solve the system of these equations, these coupled equations, to calculate the probability distribution function, and from this to calculate the rates. This is more or less the theoretical scenario. I'm not going to give more mathematical details. You can go to the articles, and those who are interested, these mathematical details are described there in great details. Okay, this is the same result. I mentioned it. So the decrease of the building blocks, or the inactivated building blocks, the increase of the activated building blocks. So my arrow it doesn't work very well. And then the formation of two structures. And this is an interesting picture, which gives us information about the probability density as a function of the angle. So I eliminate all these coordinates, reaction coordinates, and only the angle remains. And so this is the probability, the density of the angles, of the orientation of the fibers. Then you see initially when time represents times and degrees of the values of the angle of orientation of the fibers. You see that at t equal to 0, all the particles, and then you have this scale of numbers. So the probability is higher when the color is darker. You see that the particles are mainly oriented towards in the region between 100 and 80 degrees, whereas when the fuel is consumed, the particles, I mean the fibers, disorient. There is no orientation of the fibers. And when we add again activator, the particles orient in a different way, right? So orient around 30 degrees, right? Yeah, like here, you see? Okay. These are snapshots of the behavior of the different structures. So the initial, I mean, this the red fibers corresponds to the formation of the inactivated fibers, right? These fibers give rise to the activated ones, to the formation of the gel at 100 hours. At 200 hours, you see that the number of green bars increase, the green fibers increases. So there is a formation of the structures. You go back to the like scattering experiments, you see that this takes place around 150, 80 more or less, right, which corresponds precisely to this picture, right? And then since the activator is consumed, these fibers disappear. And then at this moment, you inject more activator and you start again the process. But you see that the orientation in the first activation is different from the orientation of the fibers in the second activation, right? You change the orientation of the activation, right, of the fibers. Okay, in the second example, I'll discuss the formation of Lissagan rings. Lissagan rings are formed by interactions of two electrolytes, the E electrolytes and B electrolytes. And when they interact, they form these rings, they form these patterns. And these patterns contains particles inside, right? You can see a detail of the distribution of the particles, which the size of the particles increase for the outer layers, and is much smaller in the inner layers, the inner rings, right? So this is the effect observed in the experiments. And there's another picture over here about the same feature. So this electrolyte is injected in this area, and then it interacts with the electrolyte B, and they start to form these rings, right? These rings containing particles and divided by layers which don't contain any particles, right? Okay. This, as I said, the size of the structure, the size of these structures increases as a function of the radial position. Okay, this is the mechanism of the process. So the E is the activator, is one of the electrolytes. B is the second activator. B is disactivated building blocks. A is the activated building blocks, AN are the first order structures, and GM are the second order structures. These are the kinetic processes taking place to form the rings, right? Which are described here in more details. And these are the reaction rates of each process. Right? I'm not going to give you more mathematical details about that. I mean, this is basically the essential of the process. Okay. In the model, we use the continuity equation for the probability distribution function or for the concentration of each component, okay? And we also have to combine this equation with the energy balance, because the process is not isothermal. The temperature changes in the system, as I'm going to show you in a moment, right? So in the experiments, one observes that the difference between the diameter of the different layers, the different rings, divided by the variation of the concentration of the electrolyte, E, is proportional to -1/CE, the concentration of E, which gives rise to this formula. Okay, so this is the basic model, combining the evolution equation for the concentration and the evolution equation for the temperature, because, as I said, temperature is not a constant. It increases in the rings and it decreases in the voids, right? And this is the result of solving these equations. So we solve this equation, we find out this distribution in rings, in which the darker color corresponds to the highest concentration of precipitated structures, and a lighter color indicates absence of a structure. So the center of the cylinder is precisely located here. This is the first ring, which corresponds in the picture to, let me see, to this ring. This one, right? First, second, third, so on and so forth. And you see that there are, in this case, 9 rings, and you see that the concentration of particles, the particles are larger, but the concentration is smaller, right? Concentration is smaller. Right? Okay, this is the situation observed in the experiments, and we can also analyze you can analyze the structure, the the diameter of the structure as a function of the ring position, and then you see that how this radius varies with the distance, as I said. Okay, this is the first ring, the second ring, the third ring, so on and so forth until the last ring. This experiment, this model has been, these results have been obtained with a model in which the temperature is not a constant. But to compare with this model, with these results, what we did was to analyze the case, the isothermal case, the case in which the temperature doesn't play any role. So we only solved the equation for the concentration for the probability. And then in that case, instead of 9 rings, we observed 8 rings. So which doesn't correspond to the experiments, right? So this shows that include the temperature as a variable in the analysis is important, because if not, the experimental results cannot be reproduced. And this is the temperature. This is the temperature as a function of the rings. You see that the temperature as a function of the rings, you see that as a function of position. In fact, the position, the numbers, the blue numbers are the location of the rings, and then you see that. How the temperature increases when at the position of the second ring, for example, and decreases when it passes to the third ring, and it increases, decreases, and so on and so forth. And you see that the temperature is not a constant, and it increases because there is a chemical activity, there are reactions inside the rings, and then these reactions release heat, and then this heat is the responsible for the fact that the temperature increases. So the increase is not very important, but it exists. It could be measured. Okay. Okay. So these are the 2 examples I wanted to describe in a very simple way. And then now in the last part, what I'm going to do is to propose a criterion for the formation of non-equilibrium self-assembly structures. Coming back to the problem of the potential, how to find out a free energy functional able to characterize the structures, right? You see that, as I said, in equilibrium self-assembly, the structures emerge at minimum values of the free energy, right? And then, when you know the free energy, we can calculate the probability distribution function, which is given by the canonical probability distribution function, because temperature is taken as a constant. The problem is solved. In fact, analyzing the minima of this free energy, you can identify the type of the structures, and from the experimental point of view, you can analyze the shape of the structures. But these experiments, as I said, show there are many experiments or different experiments showing that the structures arise at a specific value of some characteristic parameters, called structural parameters. For example, in the case of hydrogels, the hydrogel structures, appears at optimal values of the maximum size of the fibers that form in them, right? In the case of crystallized structures, these structures emerge at particular values of the sphericity. So the sphericity measures how different the structure is from a spherical particle. And for chiral molecules and proteins, do it at different values of the orientation of the particles. And also for the microtubule based structures, the structural parameters is the average distance between these structures. You see that the structures appears at particular values of some parameters, and you had to choose these parameters properly in order to identify the structures. But once you had done it, it can give you information about the type of structure observed. Okay. Then another experimental feature is Now I'll consider the case to illustrate this analysis. the case of the formation of polymorphs. For example, polymorphs are particles or structures of different shapes, or enantiomers, for example, right? So the reaction, the process, the self-assembly process is starting from a precursor, and these precursors can give rise to different polymorphs, BI, right? Then we start in an energy landscape. We start from the precursor, and the precursors can give rise to well, different polymorphs, right? Then I'm going to describe this kinetic process in terms of two reaction coordinates. In terms of a potential phi, which depends on two reaction coordinates, right? One reaction coordinate is gamma, and the other reaction coordinate is eta, right? The gamma describes the degree of advancement from A to the polymorphs, and the eta describes the advancement, I mean, the transformation of the different polymorphs, one into another. Okay, so this would be the potential describing the kinetics, which depends on two coordinates, one for the transformation of the precursor into the polymorphs, and another one for the change into the transformation to change of the polymorphs, right? And we will propose an effective free energy, non-equilibrium free energy, which is equal to the reversible free energy, which is the equilibrium free energy. I am calling it now phi reversible, which I call it before G. G reversible, now it's phi, right? This is the equilibrium free energy, plus the dissipation. So sigma, capital sigma, is the entropy production multiplied by T. It gives the energy dissipated, and EC is the potential barrier dividing these wells, I mean the polymorphs, right? So this is the free energy potential I'm going to use for the description. So it's a generalization of the equilibrium free energy from which we can predict the structures formed in equilibrium. But now we will use this potential to predict the structures formed in a non-equilibrium state. Okay. Well, once we have, well, you see that this phi eta corresponds to psi eta corresponds to phi gamma eta at gamma equal to 1, right? A gamma equal to 1. So if I put here a 1, this phi 1 eta is precisely psi of eta, okay? Then once we have this potential, in the same manner we calculate the probability distribution function when we know the free energy, the equilibrium free energy. We can use this potential to define a probability P of eta, okay, with these boundary conditions, okay? And then you see that you have two enantiomers, or two polymorphs. Yeah, the ratio between P eta 1 and P eta 2 is precisely, according to this formula, right, is precisely given by this, okay? Which means that some structures are more likely than others, right? The structures, the probability of observing different structures is not the same. So you can see that in this picture that the probability of observing different structures depends on the type of structures, right? In equilibrium, we only have one, but in outside equilibrium, we can have many. So you can analyze these structures by means of the sphericity, and then you see that the sphericity changes from this to this, from the disks to spheres, to pure spheres. And this probabilities means that these structures, for example, in this case, the spheres would be more likely than the disks in this particular case. Okay. Okay. Now there are also some experiments showing that these structures emerge when energy dissipation, as a function of the structural parameter, reaches a maximum. So it happens in the ecological system, in the enzymatic system, and in gelation process, that the dissipation, the energy dissipated in the self-assembly process, as a function of the structural parameters, reaches a maximum, whereas in the other cases, for example, in the formation of Lysigan rings, in the formation of hydrodynamic assemblies, this energy reaches a minimum, right? In both cases, it's an extreme, and then what we use is this fact. So this is the formulation of the criterion that the energy dissipated T times sigma has a stream at an optimal value of the structure eta star, right? So this is the condition. Okay. So we now can develop a sigma of eta in powers of the difference between the value of the structural parameter and the optimal structural parameter. Keep the second order up to second order, and also this second expansion too, combining both of them, and that we obtain is the value of the energy related to the barrier, to the barrier, to the height of the barrier dividing the polymorphs, right? You can see here this. So this E would be the energy barrier of this barrier, right? Okay, and then once we know this, this is very curious because this is this expression looks like the expression for the configurational temperature in a liquid. The configurational temperature, you know that this is a temperature related to the interactions among particles, which has the identical expression that the configurational temperature. And then in this way, when we know E of C, we can calculate the potential delta of psi, so which is the free energy in equilibrium. The dissipation we can calculate, and also this term we could calculate if we know the dissipation. So if we know the dissipation, we can calculate this third term and the second term, and then we can have information about delta psi, right? Okay, this is and then when we know the delta psi, we go back and then we calculate the probability distribution function. So we can calculate also the probability distribution function. Well, I'll give you a couple of examples, very short, to illustrate how this criterion can be used. The first one is the criterion is to study apply the criterion to study the Lysigan rings. The eta parameter in this case is the variation of the particle size from ring to ring. Variation of particle size from ring to ring. There are different structures. You can see four of them: A, B, C, and D. And then we calculated precisely all these quantities for these structures. So delta psi, delta EC, the potential barrier for the enantiomers, the dissipation T sigma, and the free energy, the equilibrium free energy, and they are represented over here. And then you see here that we can verify that the most likely structure is this one, okay, observed in the experiments. You can see that the potential we calculated has precisely a minimum value, a global minimum, precisely at this value of the structural parameter. So this, of this structural parameter, okay. So which means that the potential is successful in predicting the observation of this structure. And then this is an interesting result. And the second interesting result is that you see that the most likely structure doesn't appear at the minimum, at the global minimum of the free energy, of the equilibrium free energy, right? The global minimum of the equilibrium free energy is located over here, and is different from this value. Okay, so this shows that the free energy, equilibrium free energy, is not useful to characterize the structures, and we had to use this generalized free energy to to see how this. What are the most likely structures formed for some given conditions? Okay. In the second example, in last example, this is my last slide. We analyzed patterns in magnetic colloids. So consider a small magnetic particle suspended on a liquid air interface, under the action, on the influence of a rotating magnetic field. So when the magnetic field rotates, as I, well, indicated in the previous picture I have shown. Yeah, here. So you have these colloidal particles suspended in the liquid, on the surface of the liquid, and then you apply the magnetic field and they start to rotate. They start to rotate, they perturb the fluid, and then the structural parameter now is the reaction at the Reynolds number. Is the Reynolds number because you induce motion in the fluid. You induce motion in the fluid, and when you increase, when you rotate faster and faster the magnetic field, the Reynolds number induced is larger and larger. Okay. In this case, the potential has this form, a similar form, and the probability distribution function has accordingly this form. We analyzed two particular cases in which experiments are available. The case formed of 10 particles and the case with 12 particles. And then you see here that there are in the Celtran region there are two possibilities, two structures. One in which the central region contains 2 particles, and another one in which the central region contains 3 particles. And if you increase the number of particles, you change the structures, and then you have 2 possibilities to having 3 particles in the central region and having 4 particles in the central region. This is the potential computed by means of a model, and the probabilities are given by the expression, by this expression, right? And this has been represented in these 2 cases for 10 particles. In the first case, case A, and in the case in the second case, case B. And you see that here that the solid cycles corresponds to experiments, the hollow cycles to simulations, and the dotted line is the results of our model. You see that the potential we proposed reproduces quite well the existence of these different structures. It can be systematically used. It could be systematically used to analyze different situations. A quite general theory, in fact. Okay, that's basically all what I wanted to tell you. You have questions or comments?
Thank you very much, Miguel. Does anyone have any questions? If not, I do have a question.
Yeah.
So first, well, I have a couple, but first of all, your gels are not genetic normal gels, they're active gels, or is it a specific type of gel? Because it doesn't really fit the picture I have of a gel, but maybe it's just my naive.
Well, no, it's a, I mean, it's not an active gel, it's a typical gel. I mean, it's an example we analyze it, but we could use it. We could use the theory to analyze other types of gels, yes.
But it's something, because normally a gel just gets arrested and it's out of equilibrium because it's arrested, but it doesn't require fuel. I mean, for example, if you think of, you know.
Well, yeah, no, I analyze this in this type of gels. You need fuel you need fuel to promote the self-assembly.
Okay, so it is a specific type of gel, which is not exactly active gel, but it's, you know, something where, all right.
Yes, yes.
Thanks. Yeah. I think Pietro also has a question. Pietro?
Yes. Well, sorry if you cannot see my camera. I don't know why, but it's okay. So Miguel, probably as you know, we are working a lot with these magnetic colloids.
Yes.
And, I like it, your talk and interest in the last part. I'm a bit curious about the experimental part. You said that there are experiments that already realize this kind of small clusters. I remember there was old work from Whiteside group where they have these millimeter-sized magnets at the water-air interface.
Yes.
It was relatively famous. They published in Nature and then several following up. So it is similar, the experimental system, or basically you take they are smaller particles or micron size. Or...
Yeah, there are small particles on top of the surface, And what they do is to apply a rotating magnetic field. And then they induce motion, they induce rotation of the particles. The particles are confined, and depending on the number of particles inside on the surface, you can induce different type of structures, which differentiate from the numbers of the central particles, or the particles located in the central region of the container. This was the experiments. I don't know the particular details of the experiments, in fact, but.
Okay, because I remember you have these magic numbers depending on how many particles you get.
Yes.
You get this kind of Wigner crystal formation where you have a certain numbers that lock.
Yeah.
The outers and so on. So I guess the particles are ferromagnetic since.
Yes.
Okay.
Yes.
So they should be relatively large.
Yes.
Okay. Thank you.
Yeah. Well, this is basically a simple example we found. So we first formulated the criterion, and I mean, looking for possibilities of applying the criterion to reproduce some experiments. We find out this one, and then we did it. But I mean, some other possibilities could also be analyzed.
Sure, Miguel. In your model, you place also hydrodynamics.
Yes.
So hydrodynamic interaction between the particle apart from the dipole.
No. I mean, when you have when the magnetic field is very strong and the concentration of particles is not so high, the hydrodynamic interactions are not so important, or are not dominant, in fact, right? In principle.
But yes, there are dipolar interactions, right? Because
yes.
There's a torque there. This should be magnetic torque, I guess.
Yes.
Okay.
Yeah.
So I think Carlos also has a question.
Yes, it's also about this last part. Very interesting talk, Miguel. I'm interested in the. So I understand that in your approach, this measure to predict self-assembly, a crucial thing is to calculate the dissipation of energy, right?
Yes.
So in this case, how do you? So it's a sigma here, right? In your.
Is the sigma? Is the sigma? Yes. Yes.
So how do you calculate it in this case of the magnetic colloids?
Well, in general, I mean, in fact, the energy dissipation is T times sigma. Sigma is the entropy production, right?
Mm-hmm.
You have to calculate the entropy production due to this rotation, right? And once you have this entropy production, you can, from this entropy production, you multiply by the temperature, and you get the energy dissipation. Well, I have another. I have a slide over here. Yeah, this one. You see that what you do is basically, when you have this, for example, this activated process, you assume that this activated process can be interpreted as a diffusion along a reaction coordinate, a diffusion process along a reaction coordinate. And then the entropy production related to this diffusion process along this reaction coordinate is given by a diffusion current along this coordinate times chemical potential gradient. So how the chemical potential varies as a function of gamma, right? Mm-hmm. And then, well, this is the entropy production, basically, right? And then the J is the derivative is proportional to the derivative of the chemical potential with respect to the reaction coordinate, and then you substitute it in here. You get that the entropy production is basically, or the dissipation is basically proportional to the mu the gamma square, and then if you know mu, you can calculate the entropy production and the dissipation. So this is the way to do it in general. In general.
Okay. But so in the case of these magnetic colloids, so what reaction coordinates and what how do you know the and so I'm I
Yes.
it's not clear to me how you calculate, so how I would calculate that the entropy production in this case. But I guess I can look at the details of the paper. It must be.
Yeah, the entropy production is in fact when you can calculate, you can see that how is dissipation when a particle rotates in a liquid, right? And when a particle rotates in a liquid, the dissipation is the friction rotational friction coefficient. I mean, the velocity is the force is the rotational friction coefficient times the velocity, and then the dissipation is the force times the velocity. Okay, so knowing the velocity, you know the dissipation if you know the rotational friction coefficient. And the rotational friction coefficient depends on the field.
Yeah. Okay. So it's like a hydrodynamic dissipation.
Yeah, it's a hydrodynamics derivation. Yes. But I don't have the details here, but I can provide you the details in other occasion.
Yeah. Very interesting. Thank you.
Yeah. Thank you.
Are there any other questions by anyone else? If not, thank you very much, Miguel, and thank you very much everyone else for coming, and go enjoy your San Jordi, and I'll see you in a couple of weeks.
Thank you very much. It was a pleasure. Bye-bye.
Thank you, Antonio. Bye.
Bye. Adiós a todos.
Adiós.
Bye.